My name is Dr. Adeyinka Gbadebo. I am your biology lecturer. From time to time, I'll be doing short videos treating topics and subtopics in biology, in genetics, in ecology. Please do feel free to subscribe to my channel, like my channel, and also click the notification button. Also share this channel. Please hit the notification button so I can get updates. In my previous lecture on genetics, I treated DNA structure. I was able to elucidate DNA structure. I just want to summarize that lecture by giving you some dimensions. That's the dimension of DNA, the dimensions you find in DNA. Okay, for a DNA, a complete coil or cycle in DNA is 34 Armstrong. Is 34 Armstrong. What do I mean by a complete coil? If you follow a DNA helix, a single string, this is a single string, and that makes a complete coil, meaning that from here to here is a complete coil, and that is 34 Armstrong. Also, the diameter of the helix is 20 Armstrong. The diameter of the helix is 20 Armstrong. What do I mean by diameter? The diameter is, the diameter of a cycle. This double helix forms a diameter. So the diameter from here to here is 20 Armstrong. And approximately 10 base pairs are on a complete cycle. Meaning that from here to here, you have 10 base pairs. 10 base pairs makes this complete cycle. 10 base pair join like bond to form this complete cycle. Also, the distance between base pairs, distance between this base pair and the next base pair is approximately 33.4 Armstrong. 3.4 Armstrong. Distance between a base pair and the next is approximately 3.4 Armstrong. Now, just for conversion, 1 Armstrong is equal to 0.1 nanometer. Since you now have the dimensions of the DNA, I just want to quickly give you the implications of understanding the structure of DNA. That is, why you need to understand the DNA structure. There are two basic reasons why you need to understand the structure of DNA, which is the implication of the DNA structure. Number one is understanding the structure of the DNA makes it easier for you to know how DNA duplicates or replicates. That is, it suggests how DNA duplication or replication takes place. That is one reason why you need to understand the structure of the DNA. And then number two reason why you need to understand the structure of the DNA is because it suggests the sequence of nucleotides that dictates the sequence of amino acid in a protein. The sequence of nucleotides tells us the sequence of amino acid in a protein on a particular gene that is being expressed. On a particular gene that is being expressed, the sequence of nucleotides on the DNA tells us the sequence of amino acid in a protein that is being expressed. Simply put, two basic implications of understanding DNA structure is to understand DNA replication and to understand protein synthesis. Since we know that the basic implication of understanding DNA structure is to understand replication and to understand protein synthesis, that brings us to the topic of DNA replication. DNA replication is simply the way in which DNA duplicates itself. Understanding DNA replication helps us to know how information in the genetic material, which is DNA, is faithfully transmitted from parent to offsprings, is faithfully replicated. It helps us to know how the genetic material is faithfully replicated or generated. It's faithfully duplicated. That is why we need to understand DNA replication, because DNA replication helps us to know how the genetic material is faithfully duplicated from one cell, from within the cell, within the nucleus of the cell, how it is faithfully duplicated. It can be duplicated during gene expression. It can also be duplicated during cell division. So DNA replication helps us to understand how this information is faithfully duplicated. Now, there are three hypotheses. On how DNA is replicated. Three hypotheses has been identified. The first hypothesis here is semi-conservative replication. Semi-conservative replication. The second hypothesis on how DNA is replicated is conservative replication. Conservative replication. And then the third hypothesis on how DNA is replicated is dispersive replication. Dispersive replication. Dispersive replication. The semi-conservative hypothesis, the semi-conservative replication of DNA. This is just the illustration for semi-conservative. The semi-conservative says that from the parent DNA, in the daughter DNA that is produced, half of the parent is conserved in each daughter DNA. Half of the parent DNA is conserved in the daughter in each daughter DNA that is produced. That is, this is the DNA that is to be replicated. It unwinds, forming two template strands, and then each of the strands becomes a template strand through which a new strand is then formed. Thus, the daughter DNA that is produced conserve the original, looks like they are half of they looks like the original DNA. Conserving each of the daughter DNA, each of these template strand conserves half of the original DNA strand. What I'm trying to say is that for semi-conservative, this is the parent DNA. In the newly formed daughter DNAs, each one of the daughter DNA retain one strand from the parent DNA. These are the two daughter DNA that are formed. Each one of them retain one strand. No, they separate here. This one is involved in base pairing and form this one. This one too becomes involved in base pairing and forms a new one. So each of these daughter DNA strand that is formed retain one parental strand, and that is why they call it semi-conservative. Each daughter DNA retain one parental strand and one newly formed strand. One parental strand and one newly formed strand. One parental strand and one newly formed strand. So the second hypothesis is conservative replication. Conservative replication is trying to explain that the parent duplex, the parent DNA is replicated in such a way that the newly formed daughter, these are two daughter duplexes that are formed from this parent DNA. Semi-conservative is saying that one daughter duplex that is formed is composed of newly synthesized double helix, consists of newly synthesized nucleotides. That is why I represented with a different color. This daughter duplex consists of newly synthesized nucleotides, whereas the other duplex conserve the nucleotides, the sequence of nucleotides that is in the parent duplex. So the third hypothesis is dispersive replication. dispersive replication. In dispersive replication is trying to explain that the daughter duplexes that is formed from the parent duplex, that is formed from the parent duplex, each of them contain only segments of the parent duplex. Like from this illustration, I try to use different colors of the board marker. You see, this is the parent duplex represented with black. Now look at the daughter duplexes. Each of them contain segments of the parent, which is the black strand, segments of the parent, and segments of newly synthesized nucleoside. Segments of the parent, the black, and segments of newly synthesized nucleotide. That is what dispersive is trying to explain. So, of the three hypotheses given on DNA replication, the one that is widely accepted is the semi-conservative replication of DNA hypothesis. This one is the one that is widely accepted of the three hypotheses. And this semi-conservative hypothesis was first given by Watson and Crick. However, Matthew Merselson and Franklin Starr were able to design experiments in 1958. In 1958, were able to design experiments to actually prove the semi-conservative replication of DNA. So, what is the experiment of they call the experiment Merselson and Starr. For short, Merselson and Starr experiment of semi-conservative replication of DNA. In summary, what is this experiment all about? Merselson and Starr were able to prove the semi-conservative replication of DNA. They explained that they were trying to explain the Watson and Crick model. They explained that the DNA, which is a double helix, the DNA is a double helix, as we know. They explain that the DNA is like a zipper, the zipper of your pant trousers. It's like the zipper of your pant trousers. That is the strands. Each strands are bound together like the zipper of a material, of a pant trousers, and that is by hydrogen bond. That is the nucleotides, which the nucleotides have nitrogen bases. The nucleotides have nitrogen bases which are attached by hydrogen bonds. The nucleotides have hydrogen bases which are attached by hydrogen bonds. These are nucleotides. They are attached by hydrogen bonds. Okay, so they were trying to explain that this double helix, they are attached and they look like the zipper of they are attached, and this attachment can be likened to the zipper of a cloth. And so, in explaining semi-conservative replication of DNA, they say that this double helix will unwind. The zip will open, separating this double helix. The zip will open, separating this double helix. Let me use the zip will open, separating the double helix. Now the zip has unwind. It has unwind on this region, separating the double helix. Remember that they are nucleotides. They are nitrogenous bases. The nucleotides have nitrogenous bases, which is unwinding. So, like the zipper of a material, it has unwind, it has unzipped, and then each of the single strands forms a template. Each of the single strands forms a template. It is upon this template that free nucleotides within a cell become bound to free nucleotide within a cell base paired with the template strand on each side. Free nucleotide, this will unwind, this DNA double will unwind to the end, and then free nucleotides will then bond to each of the template strand. Free nucleotide will bond following base pair complementarity. Following base pair complementarity, new nucleotide, free nucleotide within the cell will bond to each of the template strand following base pair complementarity. By now, you should understand what base pair complementarity is. Base pair complementarity means that A will bond to T, G will bond to C. Following this rule, base pair complementarity, and then eventually, since the template strand have been unwind, eventually a newly synthesized two double helices, this will be one and this will be another, newly synthesized two double helices, two daughter double helices will be formed from the original double helix DNA in the course of DNA replication. So what I've just explained is actually the Michelson and Starr experiment on semi-conservative replication of DNA.